So now that we know about the structure of the membrane, we're going to talk a little bit about the functions. And each of these lessons at the end of chapter 5 is going to cover a different type of transport in the membrane. The first one is known as passive transport. So there's a lot of different types of passive transport, but passive transport is any movement across the cell membrane that doesn't require additional energy input. So typically, one famous example of passive transport is diffusion. Diffusion is really the tendency of molecules to want to spread out, to take up the available space. So if you think about Disneyland being really crowded, California Adventure not being very crowded, it's a hot, sweaty day, people are going to naturally try to move to California Adventure because they want to be in more open space. And that's essentially how diffusion works as well. Molecules move randomly, but depending on what molecules the cell needs, diffusion may have a specific direction. And then at a state called dynamic equilibrium, there's basically the same amount of particles crossing in either direction. So basically, if we put, say, like orange food coloring in some water, when you first put the food coloring in, the molecules of dye are all going to be in the same area. But over time, say we had like a screen and the dye could pass through the screen, the dye molecules are going to move to the other side. And even if we didn't stir it, it's still going to move. And so the diffusion is going to go across that membrane or that screen. We could also have maybe a solute, and you could see there's a lot of solute on this side. The solute, like maybe say salt, is going to slowly move to this side, and the dye is going to move across to this way, and you can see now there's dynamic equilibrium. The same amount of particles are moving in either direction. So when we talk about the movement of substances, we usually tend to say they move down their concentration gradient, aka where there's high concentration to where there's low concentration. Concentration means amount, so moving from a high amount of particles to a low amount of particles. In order to move along the concentration gradient or down the concentration gradient, it doesn't require any additional energy. So when substances are moving down their concentration gradient, we say that is passive transport. Osmosis is another example of passive transport, and it's basically the diffusion of water across a selectively permeable membrane. So it's just diffusion, but diffusion of water. Once again, water will move from where there is a lower solute to a higher solute until the concentration is equal on both sides. So don't get confused here with the solute, because that means there's more water where there's lower solute and less water where there's higher solute. So the water is still diffusing from where there's a lot of water, less solute, to where there's less water, high solute. So if you imagine, say, like your cell has a lot of salt in it at the time, the water is going to move into your cell where there's less water, more salt. So it's going to move along the concentration gradient. And this, once again, doesn't require any additional energy. So you can see here we have a low concentration of sugar, our solute, on this side of the tube. And over here we have a lot of sugar. So the water is going to move across this membrane in an attempt to dissolve the sugar. It's going to move across this membrane over to this side. And you can see now we have more water on this side of the tube and less water on this side. Over time, that just naturally happens. It doesn't require any additional energy. And the reason why that's happening is because sugar is a polar molecule and so is water. So the water wants to dissolve that sugar. It's attracted to it because of those hydrogen bonds, and it's going to come over to this side to try to dissolve that sugar. When we talk about cells like animal cells that don't have cell walls, they need to maintain a water balance. So tonicity is the ability of the surrounding solution of a cell to gain or lose water. So in an isotonic solution, there's three types of tonicity. Isotonic solution is when the solute concentration is the same inside and outside. So water is not going to move. So for example, if you have salt or sugar in the cell, you have the same amount outside the cell. A hypertonic solution is when the solute is greater outside of the cell than inside of the cell. So let's say you put your cell in a cup of soda or a cup of corn syrup. The water inside your cell is going to want to move out of the cell to where there's less water. where that corn syrup, that sugary corn syrup or sugary soda is. So water is going to leave the cell. And if you think of a cell like a balloon, when water leaves, it's going to shrink. So that's a hypertonic solution. My freshmen like to remember that because it's usually a solution with lots of sugar or salt, and sugar makes you hyper, so they remember hypertonic, more sugar or solute outside. And then the opposite is a hypotonic solution. This might be where your cell has a lot of sugar or salt, and you put it in a cup of fresh water. Now what's going to happen is the water is going to move from the outside solution into the cell. The cell is going to gain water and it's going to expand. And so in a hypotonic solution, a cell will expand. And my students like to think hypo-hippo. Hippos are big, the cell gets bigger. So you can see here, this is an example of an animal cell, and this is an example of plant cell. In an animal cell isotonic solution, there's the same amount of water inside and outside. Cell stays roughly the same. In a hypertonic solution, say lots of sugar or soda, corn syrup, something like that, the water is going to leave the cell to the solution, and that cell is going to shrivel up. And in a hypotonic solution, say there's a lot of sugar or salt in the cell, water is going to travel inside of it, it's going to expand, and it could even break or lyse, like you see here. The plant cell actually has a cell wall because you know plants, especially with water, have to take a lot in. So it's actually pretty normal for a plant cell to be full of water, and that central vacuole helps out with that. And something called turgor pressure, which exerts a force on the cell wall, prevents the plant cell from lysing. So it's actually normal and good for your plant cell to be turgid. If it has the same amount of water coming in and out, it's what we call flaccid, but basically that's like a normal plant cell, but it's really better for it to be turgid because that means it has plenty of water. And then you can see here, when it's in a hypertonic solution, say, for example, when they put salt on the roads and places where it snows to help dry up the salt or dry up the snow and the ice, you can see water is going to leave the cell to go to where it's salty, and those plant cells are going to shrivel up. So that's one difference between plant and animal cells, and that's because animal cells don't have a cell wall.
So the last type of passive transport that we're going to talk about is known as facilitated diffusion. And if you think about what the word facilitated means, it can help you remember how facilitated diffusion works. So facilitated means to help, and facilitated diffusion is basically diffusion helped by proteins. And the proteins that are helping are known as transport proteins. These help move molecules across the membrane a little bit quicker than they would be able to move across if they didn't have those transport proteins. So there are two different kind of main types of transport proteins. The first are called channel proteins, and those ones are kind of like open sesame doors. They kind of open and create a corridor that allows a specific molecule to pass through. Kind of like in the cell membrane bubble lab when you had that perfect string and it made the perfect space for the pencil or pen to move through. Examples of proteins include aquaporins, which are used for the facilitated diffusion of water, and then also ion channels that open or close in response to a stimulus, and these are sometimes also called gated channels. So you can see here, this is a channel protein, and when the molecules are ready to come through, you can see they tend to be a little bit larger when it comes to facilitated diffusion. That channel protein is just going to open up, and those molecules are going to be able to come through. The other type of facilitated diffusion happens through what's known as a carrier protein, and these proteins never open up all the way like a channel protein does. Instead, they kind of mold and change shape with the molecule, taking it in on one side and letting it into the other. But you can see this is still an example of diffusion because the molecules are moving from an area of high concentration, where there's a lot of them, to an area of low concentration, where there's fewer. So like I mentioned, the carrier proteins don't actually ever fully open or close. They instead change their shape to let that molecule move through. And that's usually triggered by the binding of the transported molecule, and then the subsequent release of that molecule is kind of what triggers the shape. But still, there is no energy required for the facilitated diffusion to happen. So those are all the forms of passive transport. In the next video, we'll take a look at active transport.